All right. Hello, everyone. I'm Ahmed Elsherbini, and I'm a student in Dr. Elsherbini's lab at the University of Kentucky. And I'd like to start by first thanking the organizers for giving me the chance to share with you some of our results regarding the exosomal ceramide role in mediating the neurotoxicity of amyloid-beta in Alzheimer's disease. So Alzheimer's disease is the most common cause of dementia and the sixth leading cause of death in the USA. It is characterized by a few hallmarks, and one of them is amyloid-beta peptides, which is formed through the proteolytic cleavage of an integral membrane protein called amyloid precursor protein, which is shown here. After being formed, the amyloid-beta peptides keep self-aggregate to form the famous amyloid plaques, which are known to be deposited outside cells and in between neurons, leading to interference with the relay of informations, and it can also trigger inflammatory response, among other effects. While the amyloid plaques have been heavily studied, the exact extracellular toxicity mechanism of their major component, which is again amyloid-beta peptides, is not clear yet, which poses a huge knowledge gap in the field of Alzheimer's research. On the other hand, our lab has previously shown that ceramide, and particularly ceramide from astrocytes, associate with amyloid plaques outside cells, as you can see here. So as you guys know, ceramide is a critical molecule in the sphingolipid metabolism. But in the context of ED, ceramide levels have been reported to be elevated in the Alzheimer's patient serum, as well as the post-mortem brain tissues. Also, ceramide is known to directly contribute to the production of amyloid-beta. More recently, ceramide has been shown to play a key role in the production of exosomes. For those of you who are not familiar with exosomes, briefly, they are nano-sized lipid vesicles that are formed through inward budding of late endosomes here to form what's called the multi-vesicular bodies, or MVBs. Those MVBs then fuse to the plasma membrane and secrete exosomes to the extracellular space, where they can act as a mean of intercellular communication. Again, in the Alzheimer context, exosomes are reported to spread amyloid-beta and phospho-tau, which is another hallmark of the disease, implicating that exosomes in propagating the disease pathology. When it comes to ceramide within exosomes, our lab actually was the first to show that astrocytes-derived exosomes can accelerate the amyloid-beta aggregation, and this effect is diminished with the addition of ceramide antibody, as you can see here. So here's a pellet weight of the amyloid-beta alone, and you can notice that it gets significantly better when incubated with exosomes. But when you add anti-ceramide antibody to the mixture, it almost goes back to the control or the amyloid-beta alone levels. In addition to that, we also reported that when treating primary culture astrocytes with amyloid-beta, they respond by secreting exosomes that are both enriched in exosomes and also carry amyloid-beta as a cargo, which made them toxic to recipient astrocytes. From that, we came up with our working hypothesis that astrocytes are the origin of toxic exosomes. We propose that astrocytes-derived exosomes are enriched with ceramide, and they can bind to amyloid-beta extracellularly, which makes them toxic when they are taken up by neurons. In order to study our hypothesis, we use a transgenic mouse model called 5xFAD, which is characterized by progressive production of amyloid-beta. And also to study brain exosomes, we actually resorted to the blood to isolate our exosomes, knowing that exosomes can actually freely cross the blood-brain barrier. So capturing them in the blood would give us an idea about what's happening in the closed environment of the brain. As for characterizations, we used the Western blot, nanoparticle tracking analysis, and mass spectrometry for lipid composition. And then we conducted the cell-based assay utilizing into-A cell line, which is a neuroblastoma cell line, as well as primary culture neurons. In order to capture our ceramide-enriched exosomes, we utilized an affinity chromatography technique that's developed in our lab and depicted here in this cartoon. So basically, we immobilized the ceramide antibody, that is homemade antibody, on magnetic beads to capture these ceramide-enriched exosomes and the associated proteins, which allows us to further analyze and characterize them in our studies. So what we found is that FAD serum exosomes has a higher expression levels of the astrocytic marker GFAP, as you can see here. Other markers are just exosomal marker for normalization, which means that FAD serum blood contained a subpopulation of exosomes that are from astrocytes and cross the blood-brain barrier. We also found that FAD exosomes are enriched with ceramide when compared to wild-type exosomes, as you can see here from the mass spec results. We next confirmed this ceramide enrichment using microscopy. So what you see here are N2A cells incubated with either wild-type or FAD exosomes and co-labeled for ceramide in red channel and flotillin 2 in green channel, which is an exosomal marker. So after subtracting the background signal, we found that ceramide signal intensity is significantly higher in cells that took up FAD exosomes compared to the wild-type one. And this is the calculation for that. So now that we know that FAD exosomes are taken up by neurons, the question was, what is the target inside those cells? So we already know that amyloid-beta affects mitochondria and Alzheimer's disease. So we decided to investigate whether exosomes actually mediate the transfer of amyloid-beta to the mitochondria inside neurons. So we first labeled our exosomes with the vibrant CM-dye, which is a lipid binding dye, and incubated our cells with either wild-type or FAD exosomes, and co-labeled for the mitochondrial marker TOM20 here in green. So not only that we found that co-localization between FAD exosomes and mitochondria, which is shown here in yellow, but also we noticed that FAD exosomes cause mitochondrial clustering, which is confirmed by Western blot against DRP1, which is a mitochondrial fission protein, dynamin-related protein 1. So both of these, the mitochondrial clustering and the increasing of fission protein DRP1 indicates that mitochondria is actually losing or are actually losing their viability. So moving forward, we already know that one of the targets of amyloid-beta and ceramide is a mitochondrial protein Vidocquain, or the voltage-dependent anion channel 1. So Vidocquain is a excuse me. So Vidocquain is a multifunctional protein that acts as a gatekeeper for mitochondrial outer membrane, and it's usually present in a homeostasis between monomer and dimer forms. And when there is an outer stimulus, in this case it would be amyloid-beta, it actually goes through conformational changes, leading to the formation of apoptotic pore, as you can see here. I will just take a moment here to mention that almost all the experiments done by other groups to study the toxicity of amyloid-beta use concentration that is around 1 micromolar, which is by itself significantly higher than the reported physiological concentration of amyloid-beta in the cerebrospinal fluid. On the other hand, our ELISA indicates that the exosomal amyloid-beta concentration that we are using is within the femtomolar concentration. So basically millions of folds below what is used in the literature. So I think that's very significant. So in order to study whether exosomes associated amyloid-beta interacting with mitochondrial VDAC, we utilize a technique called the proximity irrigation assay, or PLA, which detects the interaction between two proteins within 14 nanometer distance. And each interaction between two proteins results in a dot, like those ones here. So using antibodies against Vidocquain and amyloid-beta, we found a 6-fold increase in the PLA signals in the cells treated with FAD exosomes compared to wild-type ones, and this is a quantification here. And also to confirm that this is happening on the mitochondrial level, we combined our PLA for Vidocquain and amyloid-beta with immunocytochemistry against TOM20. And as you can see here, they colocalize and shown by yellow here. So we next checked the caspase activation, and we found that FAD exosomes lead to significantly higher level of cleaved caspase 3, which is represented by this Western blot here and the quantification. And as a confirmation, we used FLICA assay, which detects early caspase activation events, and we combined it with our PLA for Vidocquain and amyloid-beta. And basically what you see here is that whenever a cell shows a higher number of PLA signals between Vidocquain and amyloid-beta, it also stains positive for FLICA, meaning that there is a caspase activation, which suggests a mechanism by which these exosomes can exert the toxic effect on neurons. So far what I have shown is basically about exosomes isolated from serum, but we also wanted to check whether brain exosomes have the same toxic effect as the serum derived ones, which actually was true. So using FLICA assay, we found that FAD brain tissue derived exosomes were significantly more toxic than the wild type ones, as you can see here. And when we removed the ceramide-enriched exosomes using the affinity chromatography, the effect of the toxic effect of these exosomes were significantly decreased, which was true for both serum and the brain exosomes, both of them. So finally, we injected the vibrancy and the eye labeled brain exosomes into the brain of 2 weeks old wild type mice to check the in vivo effect of these exosomes. And we found that first, they are preferentially taken up by neurons, and they are shuttled to the mitochondria, where their cargo of amyloid-beta actually interacted with Vidocquain, as noted here by the green dots, which confirms our in vitro data. So to summarize, I hope that I have convinced you that when astrocytes are subjected to amyloid-beta, they secrete exosomes that are enriched with ceramide. These exosomes associate with amyloid-beta extracellularly, and this complex is then taken up by neurons where they interact with the mitochondrial Vidocquain, leading to conformational change and increase activation of caspase activation, and then subsequently apoptosis. So for our future direction, we aim to actually interfere with this, what we call the ceramide cascade in Alzheimer's disease, first by inhibiting this association between exosomes and amyloid-beta, and the second would be to inhibit the uptake of this complex into neurons. So with that, I'd like to thank my mentor, Dr. Elsherbini, and current lab members, collaborators, and funding sources for enabling this work. Thank you so much.